Hi, my name is Ciera Gardner. I am a sophomore at the University of Maryland, Eastern Shore, majoring in pre-veterinary medicine. This summer, I worked with a small team of undergrad students under Dr. Vincenzo Ellis and graduate student Scarlett Shiflett. This project focuses on the prevalence of Borrelia Burgdorferi and Paramyscus leucopus over 1 year at a specific site. Borrelia Burgdorferi is the bacterium that causes Lyme disease. Paramyscus leucopus, the white-footed mouse, serves as a reservoir host for this bacterium. The vector is Ixodes scapularis, or the black-legged tick, which is why we collected these ticks when processing the mice. To begin, we first cleared multiple trails in our trap site, Ecology Woods, located on the University of Delaware campus. This allowed our team to have easy access to sites where Sherman traps could be set. At 8:30 p.m., we put peanut butter in the Sherman traps and placed them in areas where mice would most likely be active. At 5:30 a.m. the next morning, we collected the Sherman traps and brought them back to our processing station. Captured mice were transferred from the Sherman trap to a gallon-sized Ziploc bag. We weighed the mouse in the bag and then coaxed the mouse into a corner of the bag and held it securely with our thumb and index finger. While restrained, we took an ear punch sample, tagged both ears, and checked the mouse for ticks. The mouse was then released to the location where it was captured. In the lab, the ear punch samples were stored in the fridge until DNA extraction. To extract the DNA from the ear punch, we first used proteinase K, which digests the proteins in the sample. We vortexed the tubes and incubated them in a hot water bath overnight to ensure cell lysis. The next day, we vortexed the samples, added buffers, and pipetted them into spin columns. The spin columns are used to purify the DNA in the sample. The extracted and purified DNA was pipetted into a new tube and stored in the fridge. I tested for the presence of Borrelia DNA in the ear punches using quantitative PCR. The presence of Borrelia DNA in the ear punch would indicate that the mouse was infected. A master mix composed of a forward primer, reverse primer, Tachman probe, and other PCR components were added to the wells of a qPCR plate. Next, the DNA was pipetted in the wells of the plate as well as the negative and positive controls. The qPCR plate was placed in a qPCR machine, and after 45 minutes, the results were analyzed to determine the proportion of sample mice that were infected.
40 is a positive for both. So again, you guys can see.
Finally, the early mornings, late nights, and long hours in the lab have led us to results. In 2021, 41% of the mice in Ecology Woods were infected with Borrelia Burgdorferi. However, in 2022, 66% of the mice were infected. These results have led us to ask if the Borrelia infection increased in the mouse population. Has the tick population also increased? Hopefully, with further study, we can better understand the causes of Borrelia in small mammal populations. Thank you for watching.